fantastic speakers. One is George today, and we have another one next week, Luis Larrondo. We managed to bring him from Chile. So don't miss it. It's going to be also a very good speaker. So today I want to introduce Dr. George Bloom to all of you. George took his, or graduated from University of Pennsylvania. He got his bachelor and PhD from University of Pennsylvania, where all famous people graduate these days.
Don't blame me.
And after that, he actually went for 2 postdoctoral positions, one at the University of North Carolina in Chapel Hill, and later on Worcester Foundation of Experimental Medicine, and I think it's part now of UMass, where he spent a lot of time working and studying cytoskeleton and molecules involved with different aspects of cytoskeleton before actually joining the faculty of UT Southwestern in Texas. He stayed in UT Southwestern for 16 years, and he moved all the way from the rank of assistant professor to full professor. And it was really there where his passion for Alzheimer's disease actually grew. And he's one of those few scientists that I really like very much, in the sense that he has this approach to science that is very multidimensional. He goes from the analysis of what happened in one single cell and study everything at the cell and molecular level. And as you will see today, all the way to therapeutics. There are only a few scientists that have walked the whole path that way, and I really like that aspect. And Noah was very focused in Alzheimer's disease, so which is another thing that for many of us, sometimes it's difficult to accomplish, being just focusing one disease our career, right? But it has been the enduring time in his lab. He goes all the way using primary cultures, knockout animals, transgenic animals, and now therapeutics. So you're going to listen to that story today. You won't be disappointed. After UT Southwestern, George moved to UVA. What can I say? What can I say? We got you a T-shirt from Virginia Tech here to make sure that you have a winner.
Hey, we came closer this year than in a long time.
I don't recommend you to use that on campus. But anyway.
Well, thank you, Carla.
It's a pleasure to have you here today. He's a chair of the Department of Biology at UVA, and he's here to talk about his research to all of us today. Thank you very much for coming.
Well, thank you. Thank you for that kind introduction, Carla. Can you all hear me okay? Great.
Did you turn on the microphone?
Yeah,
Okay.
I did. Is it working?
Yeah.
Good. Okay. So, despite the rivalry on the athletic fields, it's really both an honor and a privilege to be here today. So we're in the midst of an Alzheimer's disease epidemic. About 5 years ago, it became the most expensive disease in the United States. It's the leading cause of death in the elderly. About 50% of people who are 85 years old or more have Alzheimer's disease. Last year, 2017, it cost the United States about $260 billion for everything associated with Alzheimer's disease. There are now about 5 to 6 million people in the US with Alzheimer's disease, and that number may increase as much as threefold by the middle of the century, barely more than 30 years from now, if we can't develop effective therapeutic interventions, which don't exist right now. But despite all of this, in the annals of medicine, Alzheimer's is a relatively new disease. It was first described formally by Alois Alzheimer, a German psychiatrist, in 1906, who was treating a patient, August Dieter, who was young. She was in her 50s at the time, but she had all the classic symptoms and passed away a few years after she began seeing Dr. Alzheimer. The biggest risk factor by far for Alzheimer's disease is old age. So why does this, why did this woman in her 50s have the disease? And the answer is simple. She had one of the very rare mutations that cause familial early onset Alzheimer's disease with full penetrance. Maybe 1% or so of Alzheimer's patients worldwide have these mutations, although there are dozens of such mutations. So obviously, the symptoms that August Dieter had been well known throughout human history, but what made her case special was, well, actually, I'm getting a little bit ahead of myself, okay? Forgot the order in which I placed these slides. And that's got nothing to do with the Alzheimer's disease that I'm fighting. Okay, so you're all familiar with the symptoms of Alzheimer's disease: short-term memory failure, learning and memory deficits, basic cognitive failure that just gets worse with time. And I want to show this slide just to point out that at the end of the day, this is really a disease of synapses that are not functioning properly, of synapses that are lost in neurons that are still alive, and synapses that are lost due to the death of neurons. So really, if we're going to tackle Alzheimer's disease, we have to think about anything that we can do to preserve synaptic function. And in fact, so extreme is this loss of synapses that by the end of a typical course of Alzheimer's disease, the volume and mass of the brain will have shrunk by about 30%. And that's due in large part to the loss of as much as 30% of the neurons in the frontal lobes of the brain. That's lost by neuronal death, which presumably follows synaptic loss. So that really raises the question of what causes synapses to fail and neurons to die. And obviously, there's not going to be any simple answer to that, but this is what's been driving research in my lab, particularly the latter question, what causes neurons to die? And we'll get into that very shortly. But what opened up the whole show was what Alois Alzheimer did in the early part of the 20th century, something that nobody had done before, and that was to examine under the microscope brain slices from August Dieter, compare them with comparable slices from cognitively normal people in her age range, using what at the time was some newly developed silver staining methods that you see right here, which illustrates the histopathological signature of Alzheimer's disease. The neuritic plaque, which has two components. One is a so-called amyloid plaque, which you see here as this fuzzy orange material in the center, and neurofibrillary tangles, which are these very darkly stained structures that you see in this cluster of neurons surrounding the plaque. Now, the plaque is extracellular. The neurofibrillary tangles are found inside affected neurons. And at one level, they're very similar. If you were to look at them by electron microscopy, you would see that both plaques and tangles comprise very densely packed filaments that are 7 to 10 nanometers or so in diameter, variable length. They look very similar to each other, but they're made from completely different materials. The building blocks of the plaque are these peptides called amyloid-β that I'll show you about in a moment. And the building blocks of the tangles are a protein called tau, as in the Greek letter, which is a neuron-specific microtubule-associated protein, rarely found at even modest expression levels outside of the brain, and within normal brain, found predominantly in neurons. In fact, within neurons, highly enriched in axons. So a little bit more about A-beta, or amyloid-β, which I'll call A-beta. So it's the proteolytic product of a single-pass transmembrane protein called APP, or the amyloid precursor protein, which has two degradative pathways, both of which are normal, one of which leads to the fragments shown here on the left. This is the non-amyloidogenic pathway. and on the right, the amyloidogenic pathway that creates these A-beta peptides. There are about 40 amino acids in length. I say about because so far there have been about 50 chemically distinct variants of A-beta that have been found in humans. They differ from each other based on first and last amino acid and any post-translational modifications that may be present. So I'm going to talk, for the rest of the talk, I'll just talk about all these things as being A-beta, but do keep in mind that there are many different species, and in fact, some of them may be relatively inert, some of them, others are clearly much more toxic, and it's been a real difficult challenge to unravel the different functional properties of the various kinds of A-beta. One thing that's clear, however, is that in Alzheimer's disease, A-beta is made faster than the brain gets rid of it, and hence it increases with time. Well, what about tau? There's a single tau gene per haploid genome, so 2 copies for normal people. 6 splice variants shown here. They range from 352 to 441 amino acids. In the central nervous system, there's a different set of larger isoforms that are found in the peripheral nervous system. One feature that I want to point out here is this microtubule binding repeat region. So each of these things labeled R1 through 4 are tandem imperfect microtubule binding repeats of 31 or 32 amino acids each. And obviously they're responsible for most of the microtubule binding activity. I did tell you that it's a neuron-specific axon-enriched protein, but if I showed you an immunofluorescence picture of a neuron stained for tau or expressing fluorescent tau, you wouldn't be able to appreciate the microtubule association because the microtubules are so densely packed in neurons. But if you express tau, in this case as a YFP tag fusion protein in fibroblasts, you can clearly see the microtubule localization. So until about 15 years ago or so, the prevailing wisdom was that plaques and tangles are not only signs of a diseased brain, but more importantly, are somehow the cause of the symptoms. That somehow plaques and tangles, or one of them or both of them, compromise synapses and kill neurons. Okay. Well, to appreciate the role of plaques and tangles in Alzheimer's, you really have to take a step back and think about how they're formed. So obviously, they don't materialize out of the clear blue sky. Instead, they represent the end products of macromolecular protein fibril formation. And so the smallest subunits are monomers of A-beta or tau, which then aggregate to form small oligomers. And as more and more subunits add to those oligomers, and there may be some rearrangement of the subunits, you end up with these filaments that you see in both the plaques and the tangles. Now, a preponderance of evidence that's been accumulating over the last 10 years or so, maybe a little bit longer, now I think unequivocally demonstrates that it's actually soluble oligomeric forms of amyloid and tau and maybe misfolded tau monomers that are actually the most toxic species in Alzheimer's disease. Not only are they responsible for, in fact, mechanisms that I'll show you in part at least, whereby tau and amyloid work together to compromise synapses and kill neurons. But it's even worse than that. They're actually infectious. So have any of you, or most of you, heard about prion proteins? You know, these proteins that misfold, and then the misfolded versions can catalyze the toxic misfolding of normally folded equivalents. Well, amyloid and tau are just like that. They can replicate, if you will, by the same prion-like process that the classic prion proteins use to cause things like mad cow disease and Creutzfeldt-Jakob disease and others in humans and a variety of other vertebrate species. So these forms of A beta and tau are both toxic and infectious. And one of the things that we've learned in my lab, we were not the first to shed light on this, but I think we've probably done more than any other lab to advance the idea that amyloid and tau work together, as I said before. And that's sort of summarized very in a rather superficial way in this cartoon, which illustrates that there's all sorts of factors that can lead to a beta buildup in the brain, which is necessary but by no means sufficient for Alzheimer's disease. In order for Alzheimer's disease to be manifest, there has to be some kind of signaling from amyloid to tau that leads to synaptic dysfunction, neuron death, which together account for the impaired memory and cognition of Alzheimer's disease patients. So a number of years ago, one of my grad students, you'll see a picture of him in a moment, former grad student, came to me one day and said he was having trouble with his thesis work, which, and was getting very frustrated. I'm sure that doesn't apply to any of the students here in the audience. And he literally came to my office in despair one morning and said, George, I can't do this stuff anymore. I gotta have another project. And I'm really interested in neuronal cell cycle reentry. So, as you probably all know, fully differentiated neurons are permanently postmitotic, presumably in G0. But there's an extended period of time in Alzheimer's disease, maybe 10 years or longer, when many neurons in affected brain regions show all sorts of signs of going back into molecular markers of being back into the cell cycle, at least as far as through S phase. But there's no evidence that these cells ever divide. Instead, they die. Apparently, after a long delay of a year or so following reentry into the cell cycle. And it's been estimated that as much as 90% of the neuron death in Alzheimer's disease follows this period of cell cycle reentry. And although there's no formal proof yet of a mechanistic link between cell cycle reentry and neuron death, I have a very strong hunch that there is such a connection, and we're actually working on that very avidly in my lab right now. I think it has to do with failure to repair double-stranded DNA breaks that accumulate over time and eventually cause the demise of the neuron. But you know, if Carla or somebody else will invite me back in a couple of years ago, maybe I'll be able to tell you more about that. So the grad student in question, Matt Seward, wanted to take a crack at figuring out what the molecular mechanism of cell cycle reentry is all about. So the first thing that Matt did was to try to reproduce some data that were shown in this paper by Varvel et al., and then take it from there. So shown here on the left is what got us started. Primary mouse cortical neurons grown in culture and then exposed for the last 24 hours that they were alive to A beta, either monomeric or oligomeric A beta. We also exposed the neurons to fibrillary A beta, the type of A beta that you would see in plaques, but those data aren't shown in here, but the results are the same as vehicle or monomer. And during that 24-hour exposure to amyloid, we also included bromodeoxyuridine, BRDU, which gets covalently incorporated into DNA as a part of DNA synthesis. So BRDU positive nuclei are nuclei that have undergone a great deal of DNA synthesis, presumably as a result of being in S phase. So we then fixed and labeled these cultures with antibodies to BRDU and to MAP2, which is a neuron-specific protein, and we did that because there are lots of glial cells in these cultures also. And as you can see, the oligomeric, but not the monomeric A beta, nor the fibrillary A beta, cause tremendous amount of neuronal cell cycle reentry. So that confirmed what Varvel et al. Had shown. And on the right is the first really big discovery that we made in this area, and that has guided us ever since. And that's the same experiment using neurons that came from tau knockout mice. And as you can see, there's no cell cycle reentry. So this amyloid oligomer-induced cell cycle reentry is tau dependent somehow, at least in vitro, and in vivo as well. So these are tissue sections from J20 mice, which overexpress mutant human APP. The APP mutations, in this case, are fully penetrant for Alzheimer's disease in humans. By the time these mice are 6 months old, their brains are full of plaques, and they have already severe learning and memory deficits. And as you can see, lots of nuclei, neuronal nuclei, that are positive for cyclin D1, which is a G1 marker of the cell cycle. Same experiment done in J20 mice that have been crossbred with tau knockouts, homozygous for APP, null for tau, no cell cycle reentry. All of this, these little sort of red puncta that you see simply represent lipofuscin, which is highly fluorescent in every channel. And when we did these experiments, we didn't know how to get rid of that fluorescence. We now know how, but don't confuse this fluorescence with cyclin 1 positive. So even in vivo, tau is required for amyloid-induced cell cycle reentry. As an interesting sidelight, Eric Roberson, who was our collaborator for these experiments, had shown a few years earlier in a science paper when he was a postdoc at UCSF in Leonard Mookie's lab, that these tau-null J20 mice lack, don't have any of the learning and memory deficits that the parental J20 line with tau has, despite the fact that their brains are full of plaques. Okay? So what do you do with this kind of result? Where do you go next? Well, we decided to interrogate protein kinases by treating cultures with kinase inhibitors. And we found 3 kinases which, if inhibited,
George,
would...
Quick question before you move to the kinases. So when you measure do you measure the length of the S-phase before the cells become microtubule?
No.
Because...
And by the way, I didn't say they become apoptotic. We don't think that apoptosis is the principal death mechanism.
That was the other question.
Okay.
Thank you.
In fact, we have some evidence suggesting that it is not. I mean, that's not to say that some cells will, but there's, in Alzheimer's disease in general, some of the animal models, particularly fly models, Drosophila models of human Alzheimer's, there's a lot of apoptosis. But there's not much evidence for widespread apoptosis in human Alzheimer's disease. Okay, so anyway, so inhibitors of CAM kinase 2, FIN, which is a non-receptor tyrosine kinase, and protein kinase A, or cyclic AMP-dependent protein kinase, could block cell cycle reentry. And, but interestingly, one of the principal tau kinases, GSK3-beta, when inhibited, did not block cell cycle reentry. That's not to say GSK3-beta isn't important in Alzheimer's disease. It says, at the very least, that it's not important for this cell cycle reentry response. Matt also reproduced a result that was published in that Varvel et al. Paper, namely that rapamycin, which is an mTOR inhibitor, could block cell cycle reentry. And I'm going to talk a lot about mTOR shortly, but in the meantime, to try to take this one step further, we thought, well, maybe maybe amyloid-beta oligomers are working through receptors that talk downstream to mTOR, principally insulin receptors and IGF-1 receptors. And so we inhibited those and had no effect, and we dropped the mTOR for a little while, but as you'll see shortly, we got back to it. But anyway, the bottom line of Matt's paper is shown here, which is basically the outlines of a crude outline of the cell cycle reentry network, whereby amyloid oligomers, by some mysterious processes, activate a bunch of protein kinases, which then phosphorylate tau at specific sites. I didn't show you the data for that, but each one of these sites is absolutely required for cell cycle reentry under the control of the indicated kinase. And then by some mysterious processes, this all leads to cell cycle reentry. Okay. So Matt finished this up, went on to take a job at Eli Lilly and Company. And then Andres Norambuena, who's now a senior scientist in my lab and is an outstanding cell biologist and neuroscientist. And if there's any faculty in the audience looking for a job, a real job, came in and took over this project and really took it to the next level. And this shows about 90% of what we now understand about this signaling network that controls cell cycle reentry and neuron death. If I had 3 hours, I could show you the evidence for all of this stuff. But what I'm going to do instead is just focus on one part of it, which is this connection between tau and mTOR. So what is mTOR? Just out of curiosity, who in the audience has heard of or knows anything about mTOR? Okay, good. So now we're going to have a little quick mTOR 101 lesson to bring everybody up to speed. So mTOR is a serine threonine kinase, but unlike most kinases, in order to be functional, it must be associated with membranes and also be part of either of two multi-protein complexes called mTORC1 and mTORC2, which share some subunits but are discriminated from each other by unique subunits that are unique to each complex, in particular Raptor for mTORC1 and Richter for mTORC2. There are also some well-known drug inhibitors of mTOR. I already showed you rapamycin. It works by competing with Raptor for binding to mTOR. And since Raptor is required to hold the whole mTORC1 complex together, rapamycin causes the dissociation of the complex and by extension inactivation of the kinase activity. Indirectly, it affects mTORC2 activity, but it takes a lot longer to inhibit mTORC2, and I'm not really going to get into that. Okay. Okay, so this is what mTOR does. Now, I understand that the graduate students here are required to sign in, and this is actually a formal course. So take a careful look at this, because there's going to be 3 test questions about this figure when I'm done. Okay. Basically, mTOR is involved in just about everything that happens in cells. It's a master regulator of cellular behavior. How does it work? It's close downstream to extracellular nutrients like amino acids and glucose, and extracellular trophic factors like insulin and a variety of growth factors. And so when those things are abundant, mTOR gets activated. And when they're not abundant, mTOR is silent. Okay? So what happens when mTOR is activated? Well, for one thing, it's mTOR, well, amino acids and glucose can activate mTORC1, which then leads to all sorts of downstream effects: protein translation, cell proliferation, cell survival. Okay? And when it's not activated, namely when there isn't a lot of food or trophic factors around, another thing that happens that isn't shown here is mTORC1, when it's activated, inhibits autophagy, which is good, because you don't want to be eating yourself when you're surrounded by abundant foodstuffs and trophic factors. But if you're starving, which actually is a problem with the Alzheimer's brain, as you will see, then you want to activate autophagy to provide yourself with, you want to eat part of yourself to provide nutrients for whatever's left over. mTORC2, on the other hand, is activated by insulin and growth factors, and its activation leads to mTORC1 activation, which in turn, by the routes shown here, leads to mTORC2 deactivation. So the point is that in normal mTOR signaling, the signaling is transient. mTORC1 and or 2 get activated briefly, they do their thing, then they shut off. Okay? But in Alzheimer's disease, mTOR kinase activity is constitutively elevated. Now, a lot of protein kinases can be monitored in terms of active versus inactive state by Western blotting or immunofluorescence using antibodies to phosphorylation sites that are 100% correlated with enzyme activation or deactivation. That can't be done with mTOR. There's a bunch of phosphorylation sites, but they don't affect kinase activity. So to monitor kinase activity, we have to look at net phosphorylation of mTOR substrates. And so I will show you, I think I'm going to show you some blots of 4-EBP, which is an mTOR substrate. More phosphorylation means more mTOR activation. Okay, so I told you about activation of mTORC1 on the lysosomal surface that causes an inhibition of autophagy. We already knew that mTOR was activated by amyloid oligomers and furthermore is required for cell cycle reentry. So we asked, where does mTORC1 get activated? And I say that because lysosomes are not the only place where mTORC1 is found. It's also found at high levels on the plasma membrane, although until the work I'm about to show you was done, its function on the plasma membrane was very poorly understood. Probably still is poorly understood, but now we know a little bit more about it. So what we did was we took primary neurons and overexpressed flag-tagged raptor tagged with the plasma membrane targeting domain of HRAS or the lysosome targeting region of REB. And what this does is it causes almost all of the mTORC1 to be clustered either at the plasma membrane or the lysosome. And we then challenged both types of cultures with amyloid oligomers. And we found that when you target mTORC1 to the plasma membrane and challenge the neurons with amyloid oligomers, they go back into the cell cycle. But when you force all of the mTORC1 to the lysosome, there's no cell cycle reentry response. So this is an unconventional mechanism for mTORC1 activation because to the best of my knowledge, every other thing that has ever been looked at in terms of activating mTORC1 leads to lysosomal activation. In many cases, the plasma membrane has not been looked at, but in this case, we don't think there's any lysosomal activation. It's all at the plasma membrane, and so that is necessary for cell cycle reentry. Furthermore, we found that tau is required for this preferential activation of mTORC1 at the plasma membrane. So here we're looking at 4-EBP phosphorylation in wild-type and tau knockout neurons in the absence and presence of amyloid. And so this is total 4-EBP. This is total 4-EBP. This is the phospho version. And nu-N is just a neuron marker. And what you can see is that in both wild-type and knockout cells, we do get statistically significant activation of mTORC1 by amyloid oligomers. But when we do the experiment, when we modify that experiment as I described in the previous slide, namely forcing mTORC1 to either the plasma membrane or to lysosomes, we find, as shown earlier, preferential activation of mTORC1 at the plasma membrane in response to amyloid oligomers in wild-type neurons, okay, but not in tau knockout neurons. So there's something about tau that transduces the signal from amyloid-beta to mTORC1 that causes that activation of mTORC1 at the plasma membrane. That absolutely requires tau. We don't understand the mechanism, but we're very anxious to figure it out. So why would mTORC1 be important? Well, we know that Tau is phosphorylated in Alzheimer's disease at serine 262. In fact, that's in one of the microtubule binding repeats, and it can partially compromise microtubule binding. We don't think that that phosphorylation is under the direct control of mTORC1, although it might. It's hard to prove formally. But we do know that one of mTORC1's substrates, another protein kinase called S6 kinase, becomes phosphoactivated by mTORC1 and can phosphorylate tau at this site, both in vitro and in vivo. Okay? So we began looking at serine 262 phosphorylation on tau, and this is a well-known Alzheimer's model mouse, triple transgenics, made by Salvatore Oddo. And Salvo's lab took these mice, and from the time they were 3 months old until they were 18 months old and sacrificed, they were given ad libitum access to rapamycin-laced food. And as you can see, that led to a big decrease in tau phosphorylation at serine 262. And in a separate model, TG2576, this is another Alzheimer's model, they made a genetically engineered variant that was haploinsufficient for mTOR just in the brain. And as you can see, those animals that have half doses of mTOR have much less phosphorylation at serine 262. So all of this shows correlation between serine 262 phosphorylation, mTOR activation, and amyloid insults, but it doesn't prove anything about serine 262, but this does. So if we take wild-type neurons, treat them with amyloid oligomers, you get a lot of neuronal cell cycle reentry. Okay, excuse me, I'm getting ahead of my. Okay, I misstated this. These are tau knockout neurons in which we re-express human tau using lentiviruses. So if you re-express wild-type tau, you can recapitulate the response, the cell cycle reentry response to amyloid oligomers. But if instead you overexpress equal amounts, or even a little bit more, tau that's been mutated to an alanine at position 262, so it can't be phosphorylated, there is no cell cycle reentry. So that site, that represents a fourth phosphorylation site under the control of mTORC1. So at the very least, that's one of the reasons that mTORC1 is required for this phenomenon.
And even with that mutation, they form oligomers? Even with the 262 mutation to alanine, still tau forms some kind of oligomer?
You know, we've not looked at that, but it's a good question. And it may well be that that's why they're not responsive. We're actually doing some experiments along those lines, but they're in progress, and so it's not something we don't have the results yet. Okay, so if you think about this, what I've just told you about is a toxic positive feedback loop between between mTORC1 and tau. So tau must be phosphorylated at serine 262 under the control, direct or more likely indirect, of mTORC1. And at the same time, tau, including phosphorylation of tau at this site, is apparently required for the mislocalized activation of mTORC1 at the plasma membrane. So because of that, as well as something else that I'm going to show you in a few minutes, we're beginning to think that whatever else Alzheimer's might be, it should be thought of as an mTORopathy, because it represents a crucial part of the Alzheimer's equation is fundamental dysregulation of normal mTOR activity. Okay, so now I want to move on to the second story. I'll be very brief about this. Just published a couple of months ago in Alzheimer's dementia by a former grad student who just completed her PhD, Erin Kotis. And I want to talk about this arm of the signaling network, particularly calcium. So this is really the bottom line, the last figure in Erin's paper. I'm just going to show you two more figures after this. And it summarizes everything she learned, which is that amyloid oligomers cause an excess calcium influx through NMDA receptors, and at least in part because of the consequent activation of CAM kinase 2, calcium calmodulin activated protein kinase 2, that leads to cell cycle reentry. In fact, this is absolutely required. So we know that these NMDA receptors are essential for this because of if we knock down essential NMDA receptor subunits, we prevent cell cycle reentry. If we chelate excess calcium globally with BAPTA, which is a sledgehammer, we block cell cycle reentry. And then there are also pharmacological approaches, drugs that specifically inhibit or antagonize the NMDA receptor block cell cycle reentry. So one of those drugs is Memantine. And it's a very interesting drug because it can discriminate healthy from malfunctioning NMDA receptors. So you might think of the NMDA receptors. The NMDA receptor is a ligand-activated calcium channel. You might think of it as being in a closed or an open state. And when things are working properly, ligand binding, neurotransmitter binding to the receptor causes transient opening of the channel. Just the right amount of calcium comes in. And if there's enough receptors that are involved in this, you generate an action potential. Everything works perfectly. But amyloid oligomers, by a tau-dependent process that I won't get into, cause the channel to remain open for an excessive period of time that then lets excess calcium in. And that can be acutely synaptotoxic. It can destroy synapses, maybe permanently, at higher calcium influx, or in extreme cases, even cause acute neuron death. So Memantine can actually easily get into the channel of a receptor that's open for too long, but has a very small chance of getting into a channel that's opening and closing properly. It binds reversibly. So if it does accidentally get into a normal channel, it'll dissociate pretty quickly. The channel will be fine. But in the case of a malfunctioning receptor, if it dissociates, it'll be quickly replaced by another Memantine, which will continue to prevent excess calcium entry. Okay? So in culture, Memantine very effectively blocks cell cycle reentry, and in vivo as well. So here we're looking at TG2576 mice again that were treated. We know that when they're 5 weeks old, 3 months old, that they have abundant cell cycle reentry. And if you add to their water Memantine when they're 3 weeks old at weaning and let them have ad libitum access to Memantine, which yields steady-state plasma concentrations of Memantine that are known to be therapeutic in humans, and I'll get to that in a moment, we can completely block cell cycle reentry. So why is this important? Well, Memantine is one of 5 FDA-approved drugs for Alzheimer's disease. Now, I told you at the beginning that we don't have any effective drugs, and I'm not contradicting myself now. These other 4 are all cholinesterase inhibitors. They're Band-Aids. They take cholinergic synapses that are barely functioning, and by allowing the acetylcholine to stick around for longer than it would otherwise, they increase the chance that a damaged but still functioning neurotransmission can take place, at least for cholinergic neurons. And so when these drugs work, they work very modestly for short periods of time, but they're no panaceas. I told you about Memantine. It's the last of the drugs to be approved, but it's only approved officially for moderate to severe Alzheimer's disease. And that's because you can see benefits in many, but by no means all patients at that disease stage. But nobody's ever tested it pre-symptomatically. So as a result of what I just showed you, we're actually now at the early stages of designing a clinical trial, or maybe even a couple of related clinical trials, to test the hypothesis that Memantine has disease-modifying properties, that it might be an effective prophylactic. And so if I'm still around and active 5 or 10 years from now, I'd love to come back and tell you about the results of that trial. But it's going to be a very lengthy trial because it's going to be analogous to the way that statins are used to protect against cardiovascular disease. You identify patients at risk, you begin treating them long before they become symptomatic in the hope that that will prevent them from ever becoming symptomatic. In the case of Alzheimer's patients, we're probably going to go with people who are positive for the epsilon 4 allele of the apolipoprotein E gene. That allelic variant is a very strong risk factor for Alzheimer's disease. Okay, so putting this all together about cell cycle reentry, there's another thing that I want to point out. I haven't really talked about it, but I'll spend a minute or so on this, and that is insulin signaling. Now, one of the big breakthroughs that we made in understanding this cycle, or in this network, was that you could block cell cycle reentry by adding insulin to the medium in which the neurons are growing. And you might think, aha, that's the cure for Alzheimer's disease. If you could somehow get insulin specifically into the brain without without delivering it systemically, because that could cause all kinds of problems, maybe you could prevent Alzheimer's right from the beginning. Well, the problem is that the Alzheimer's brain is characteristically insulin resistant, irrespective of whether or not the patient has systemic type 1 or type 2 diabetes. And that insulin resistance in brain is caused by amyloid oligomers, which initially lead to a sequestration of insulin receptors into the cytoplasm, presumably by endocytosis. So they're there, but they can't sense the insulin. And then at later disease stages, there's a huge loss of insulin receptors at both the messenger RNA and the protein levels. So the ability of amyloid oligomers to impair insulin signaling in neurons then unleashes the further toxic potential of those amyloid oligomers to drive neuronal cell cycle reentry and neuron death. The other thing I want to point out before moving on to the final chapter of the story is that, at least in vitro, the kinetics of cell cycle reentry are very rapid. Within hours of exposing neurons to amyloid oligomers, they're already deep into the cell cycle. And this occurs by a mechanism that's initiated by amyloid, proceeds by a tau-dependent manner, completely independently of the incorporation of amyloid and tau into plaques and tangles. And so when I told you at the very beginning that it's the soluble oligomeric forms, or maybe misfolded tau monomers, that are the real bad guys, this is one of the key pieces of evidence. Okay, final chapter. Just out a couple weeks ago in the EMBO journal, along with a News and Views article about the paper. Another beautiful body of work masterminded by Andres Norambuena. I just sit in my office and type on my computer all day long. Andres and the others do all the work. This is a story of another amyloid tau story, but a completely different direction, beginning with the coenzymes NADH and NADPH, which of course are respectively involved in glycolysis and oxidative phosphorylation for NADH, and for NADPH for biosynthesis. Okay. Have you ever done immunofluorescence of tissue culture cells and maybe had an unstained control, and you look at it on the microscope and you see this faint fluorescence that looks like mitochondria? Well, it is mitochondria. And that's because NADH and NADPH are intrinsically fluorescent, not very brightly so, but intrinsically fluorescent. And in fact, as shown here, the intrinsic fluorescence, which you can see with the, all you have to do is the right excitation and emission windows on your microscope, shows fluorescence that co-localizes almost perfectly with a mitochondrial marker. Well, so you can use NADH, NADPH fluorescence as a tool for what's happening in mitochondria, but there's a lot more to it. And that is that the fluorescence lifetimes of these enzymes is affected dramatically based on whether or not they are free in solution or bound to enzyme partners. And there's a big increase in the fluorescence lifetime, on the average about 4-fold, when an NADH or an NADPH is bound to an enzyme partner. And you can see here, this is sort of a distribution of lifetimes. So this is 2-photon fluorescence lifetime imaging of live neurons. And this is a distribution of lifetimes for everything that you see here, which is color-coded to indicate this A2 percentage, just basically another way to express the lifetime. Okay? So we also know that, so taking advantage of this, Andres discovered something brand new under the sun that he calls nutrient-induced mitochondrial activation, or NEMA. So this is a very fundamental cell biological phenomenon. And the only thing that amazes me about it is that nobody ever saw it beforehand. So you take any kind of cell, in this case neurons, but we've done this in other cells also, and you basically starve them of nutrients. You put them in a balanced salt solution just for a short while, and then stimulate them with either nutrients like amino acids or trophic factor like insulin. And what you see within minutes is a big shift in the lifetime of these coenzymes. And so basically each one of these colored curves represents, with the solid lines, represents before stimulation, and the curve with the dotted line of the same color illustrates the same field of view after stimulation. And what you could see is statistically significant shifts of life to longer lifetimes after stimulation with both amino acids and insulin. So in other words, these things quickly lead to stimulation of mitochondrial activity. And it requires mTORC1 activity, not surprisingly, because I already showed you that things like insulin and amino acids stimulate mTORC1. So you may recall that Raptor is an essential subunit of mTORC1, and if we knock that down with siRNA or shRNA and do the same experiments, we see no NEMA. So mTORC1 is required. And furthermore, it's required at lysosomes. And we know that because if we knock down either of two constitutive inhibitors of mTOR that are located on lysosomes, we prevent we prevent NEMA. Okay, so this is in contrast. This is the opposite of what we saw for cell cycle reentry. This requires mTOR activation at lysosomes, and furthermore, it's blocked by amyloid oligomers. But the amyloid oligomers do not block NEMA in tau knockout neurons. So just like cell cycle reentry, tau is required for the inhibition of NEMA by amyloid oligomers. And that's because amyloid oligomers activate plasma membrane mTORC1, but not lysosomal mTORC1. But remember, we are stimulating these neurons with insulin or amino acids, which does stimulate lysosomal mTORC1. But that stimulation is trumped, I hate to use the word, but by activation of plasma membrane mTORC1. That overrides. And that's what's, and so a summary of NEMA is shown here. Normal cells, insulin and amino acids activate lysosomal mTORC1, which then lead to mitochondrial activation. And this is tau independent. And by the way, the other new thing under the sun is this functional connection between lysosomes and mitochondria that nobody had ever seen before. But in Alzheimer's disease, amyloid oligomers inhibit NEMA and promote cell cycle reentry. So, and they both occur because of the activation of mTORC1 at the plasma membrane in response to amyloid oligomer exposure. Okay. So putting this all together in a grand scheme, the final slide that I'd like to show you, which is the title of the talk, is that amyloid is the trigger and tau is the bullet for Alzheimer's pathogenesis. That amyloid, somehow or other, bad amyloid makes good tau go bad, and then bad tau does all kinds of bad stuff for cells. And I showed you about NEMA, I showed you about cell cycle reentry. I didn't talk about synapse. Same holds true for synaptic function. That's mostly work done by other laboratories. But again, as I said just a couple of minutes ago, I sit in my office typing on my computer most of the time. And these are some of the people who've done all the work, as well as additional work that I didn't have time to tell you about. This is an old picture from the lab. We're in the midst of updating our website and making new pictures. Hopefully that'll be up pretty soon. You've heard the names of some of the principal people behind the work, but there are lots of others, including collaborators and our generous funders. So Carla said to be done by 1:15. It's 1:17. I apologize for being 2 minutes over, but I'd be glad to answer any questions you may have now.
Nice story on your cell cycle reentry. So DNA viruses, when they replicate in cells, they have to activate cell cycles so they can access RNA. Do you think, is there any virus, DNA virus that is involved in Alzheimer's?
Well, there actually have been, there's a recent report from Rudy Tanzi's lab about, I believe it's herpes virus, that when reactivated can cause, can sort of nucleate the formation of amyloid filaments. And although I told you that those filaments are less toxic than the oligomers, when they reach a certain concentration, they represent reservoirs from which toxic oligomers can diffuse away. So there's a lot of interest in that idea right now. Now, I don't know if herpes is a DNA virus, right?
Yes.
Yeah. So so there's a lot of interest in this. I think my personal feeling is that that herpes and maybe other viruses can be contributing factors. In some cases of Alzheimer's, maybe they're the main thing that gets it started. But I would be very hesitant about concluding that it's a universal mechanism for initiating Alzheimer's disease. I could be wrong, and if so, the evidence will prove it, but that evidence isn't here yet.
Next talk. In your model, you suggest that tau must be protected with 4 different sites, correct?
Well, at a minimum, there may well be other sites that are just as essential that we haven't uncovered yet.
And do you tau is a consequence vector for that model, and if it's true, then what happens is that tau is actually being added for multiple negative charges on the surface that can induce oligomerization of the protein that triggers cells activation. Maybe it's too much speculation, but can you?
No, that's exactly my thinking. In fact, you could have been an audience plant for a question that the speaker would have liked to have had asked. So what I didn't tell you about tau is it is an intrinsically disordered protein. It does not have any stable alpha helix or beta sheet structure. However, it does have some fairly stable tertiary structure in the sense that the N and C termini reside very close to each other, like less than 2 angstroms apart. And furthermore, about 2 angstroms away from the middle of this microtubule binding repeat region. And it's this form of tau that we think favors tau binding to microtubules and but does not favor binding of tau to other tau molecules. Another thing I didn't tell you is that the interaction sites on tau for other tau molecules are within the microtubule binding repeats themselves, particularly the 2nd and 3rd repeats. So a tau molecule can either bind to a microtubule or another tau, but not to both at the same time. And we think what's happening in Alzheimer's disease is that tau, either by post-translational modification along the lines that you said, okay, and or proteolysis that cleaves some or all of the N or C termini, opens up the molecule and exposes this microtubule binding repeat region in a different way that now favors tau binding to other tau molecules. So take this one step further. The 4 sites that we've identified are all very close to each other in this so-called hairpin or paperclip normal conformation of tau. So we've been thinking exactly along those lines, that this high concentration of negative charges due to phosphorylation might cause the unfolding of tau and convert it from a normal form to a toxic prion-like form. We're in the midst of doing experiments to test that hypothesis, but we don't really have the answers yet.
Can you switch to lysine residues?
Pardon me?
Can you switch these amino acids to lysine residues and do the opposite?
Yeah, we could. We just haven't done that yet.
So do you see your phosphorylated tau mechanism in cell cycle reentry in other diseases that have
I'm really glad you asked that. It's a very common, if not universal, feature of every major neurodegenerative disorder, including Parkinson's disease, frontotemporal dementias, traumatic brain injury, CTE, Huntington's disease, ALS. And one of the things I'd like to do, but we haven't really, you can't do everything you want to do, but if in a perfect world, I would love to interrogate this pathway in all of those diseases. Not so much from the amyloid standpoint, because there's probably no reason to think that amyloid is involved in any of these other diseases, but at least as regards the tau involvement.
Yes, please.
Regarding the treatment and at least prevention of Alzheimer's disease in that direction, I'm learning that the ABOS oligomers are found outside the cell. They're extracellular, mostly. They are separated as monomer, they accumulate, and they are stuck as oligomers outside the cell.
Yes, but there are intracellular oligomers. They've just been almost ignored.
Right.
Yeah.
But still, I'm wondering if there is a way to increase the fluid circulation to the central nervous system by mechanical means, physical means, some way to increase the fluid circulation by promoting the lymphatic circulation, for example.
Yeah.
Along the line. If there is a way, then we should be able to break down the ABOS plaques into smaller pieces, which is more readily excretable from the central nervous system, like using physical phenomena like this sonic waves or sound waves that can go on, hit those little plaques, making the small ones, ultrasounds, for example.
Yeah.
Do you have any thoughts on that direction?
Yeah, I do. I would be very surprised if breaking down plaques into smaller things would help. I think it would have the opposite effect because you would be generating a lot more toxic oligomers that way. On the other hand, you did talk about the glymphatic system, and one of my close colleagues at UVA, Yoni Kipnis, who's chair of the neuroscience department, has published a couple of papers showing, well, initially, the discovery of lymphatic vessels in the brain meninges, which violated everything that was ever taught in neurology textbooks, which claimed that there's no lymphatic system associated with the brain. And he's also shown more recently that those lymphatics, which are connected directly to the ventricles in the brain where toxic forms of amyloid and tau can accumulate, actually flow out of the brain through these lymphatics back, presumably eventually into the general circulation. And in an experimental system, he showed using some Alzheimer's model mice that those lymphatic vessels tend to constrict in Alzheimer's, at least in the mouse models, and that if you can dilate them, you can flush out of the brain some of these toxic forms of amyloid and improve outcome for the mice. So I agree in part with what you said, but I'm skeptical about breaking up plaques. I think that would be bad, but I could be wrong.
Hypothesis is that in the aging brain there is a reduction in fluid circulation. That's normal. But aging is not a disease. We all want to live long. Right.
Well, up to a point, maybe.
Right. So by some means, we can increase the circulation in the aging brain. That might facilitate the clearance of ABOS.
Yes, absolutely. I mean, even cerebrovascular, blood. I think there's a really big role for cerebrovascular impairment in Alzheimer's disease, and there aren't a lot of labs that are looking at that.
All right, thank you.
Okay.
Any questions from the students? Yes.
Would an mTOR inhibitor in conjunction with Namenda hurt or help?
So that's a great question. Salvatore Otto, who's our collaborator for those experiments, has published a good handful of papers on treating mice with rapamycin. And he finds, it reduces the plaque and tangle burden, it improves their behavior. It doesn't really make a difference on lifespan because the mice live as long as, at least the ones he's been looking at. But there is quite a reluctance to translate that directly into the clinic. Rapamycin is an FDA-approved drug, but it's got some nasty side effects. It's used in cancer chemotherapy, but only in, reluctantly in very selective cases. There are drugs, they're so-called rapalogs, variants of rapamycin itself, quite a number of them that may have, that are safer. I don't know if any of them are FDA approved yet, but that's a potential avenue. But you got to be, my feeling is if you go after enzymes that are ubiquitously expressed and are involved in fundamental aspects of cell biology, you're going to be damn lucky if going after that kinase fixes your disease without causing other problems. So I'm more inclined to go after strategies that be more specific for Alzheimer's disease. I think tau is a very good target because anything that you do to interfere with tau is very unlikely to have any side effects, because tau is basically restricted to the brain and the peripheral nervous system. So you could have effects on peripheral nervous system tau. And there are a number of labs, working on both passive and active tau immunization, small molecule inhibitors of tau oligomer formation, and so forth. Those are all pretty early stage still.
I'm going to have the very last question. Sorry. It comes to the treatment, your idea of a prophylactic treatment for this disease, for those that they have the chances of developing the disease. Besides all the ethical issues or whatever that we can discuss or not, the point is, how early would you need to start? Because I guess if you start when you have symptoms, or early symptoms, it might be already too late.
Yeah. So
yeah.
If
you start when there are no symptoms, someone might argue that they might never develop the disease at all.
So my personal opinion, which I've been arguing for at least a few years, and I would say just within the past year, there's been a sea change in the field, among leaders in the field about this. You're absolutely right. The new consensus is that we should not hold high hopes for doing much to help patients once they become symptomatic, by treating them, beginning to treat them, however you treat them, once they become symptomatic. That the ultimate answer is going to be early intervention, pre-symptomatically. So we have to be able to identify patients at risk. ApoE4 is a good hint, but maybe we can do better than that. And we need to be treating them with something that can prevent all of the stuff that I just told you about from ever getting started. And right now we know everything we need to know to do that, except how to identify those people and what to give them. But we're working on it. No, I mean, I think there's a lot of hope for that in the future. And much of that, the field has been dominated by the idea that if you could only get rid of plaques and tangles or prevent them from forming in the first place, that would fix everything. I hope I convince you that that's an extremely naive view, and that we really need to delve much deeper than I showed you into the basic molecular mechanisms that are behind the disease as a prelude to developing effective therapeutics, whether they're preventive therapeutics.
Okay, guys, we need to stop here. Thanks, George. All of you can sign up for lunch. Please wait here. We're going to take it to the room for lunch. One more question. So this is a genetic disease.
Not usually. I mean, so the presenilin and APP mutations may be 1% of patients worldwide. ApoE4, maybe 50% of cases, but it's not fully penetrant. It's just a big risk. In fact, I've even heard, there are even some statistics that say if you're ApoE4 positive and, well, all other things being equal, if you're ApoE4 positive, one copy, about a 5-fold higher chance of becoming symptomatic if you live long enough, maybe 10 to 20-fold if you're homozygous. But if you're asymptomatic by the time you're 75, you'll probably never become symptomatic if you're ApoE4 positive. Yeah, I just got a few more years to go.
Say hi to John and Deb.
Yeah, will do. Okay, nice seeing you, David. Yeah.
Friday, next Friday, is it the Emily? I have an exam on Friday.
This might sound relevant for someone
Yeah.
And also, is there some way of promoting, let's say, tau degradation, either proteasomal or agosomal-mediated degradation of tau, or even just before, preceding the tangle sort of? I mean,
So the tau knockout mice have normal lifespans and fecundity. They develop age-dependent neurological abnormalities, but you know, clearly it's not essential for life.
Uh-huh.
And you're-
Okay, we have two different strains that we keep. One of them, they spend a lot of time chasing their tails. I don't know why that is, but I don't think the other strain does it. So I don't know if it has to do with tau knockout by
That
itself.
basically lead into the second question.
Yeah.
As in